Hello, my name is Jason, and I'll be presenting on regulatory RNAs using the versatile transcriptome of bacteria as an example. So a little bit of background information to start. We first want to know what an RNA is, and an RNA is a small nucleic acid molecule present in living cells. Its principal role is to act as a messenger carrying instructions from DNA for controlling the synthesis of proteins, although in some viruses, RNA rather than DNA carries genetic information, which is important to note. There are several types of RNAs. There's messenger RNA, also known as mRNA. Transfer RNAs, which is a adapter molecule composed of RNA, typically 76 to 90 nucleotides in length, that serves as its physical link between mRNA and amino acid sequences of proteins. And microRNA, which plays a role in gene regulation, mRNA silencing, and post-transcriptional regulation of gene expression. MicroRNAs usually function via base pairing with complementary sequences within the mRNA molecules. In this presentation, we'll be focusing mostly on small RNAs, but to start, we'll look at microRNAs and small interfering RNAs. I previously described what microRNAs and what microRNAs were in the previous slide, but small interfering RNAs sometimes known as short interfering RNAs or silencing RNAs, are a class of double-stranded RNAs, RNA non-coding RNA molecules. They're usually about 20 to 25 base pairs in length, similar to microRNAs, and operating within the RNA interference pathway. They play a role by base pairing with messenger RNAs. Usually, if the base pairing is close to completely complementary. Most of those messenger RNAs are marked for degradation if complementarity isn't as if base pairing isn't as complementary. It inhibits the microRNA from being able to be translated. So the question there is, are there other small RNAs in this network with similar roles, or perhaps even different other roles? Here's a list of other gene regulatory networks we discussed in class. One of which is chromatin compaction, and how that the level of compaction often impacts how easy it is for transcription factors to access the DNA to initiate transcription of several genes. other general transcription factors or the regulatory transcription factors. Transcription itself, how RNA is processed, the spliceosome, and the role microRNAs and lentiviral RNAs play in gene silencing. Here's a figure from our textbook showing the processing of a micro or small RNA from its precursor molecule, from its precursor microRNA all the way to its microRNA stage, being exported out of the nucleus via the RNAs network of molecules. So one of the issues I found upon trying to review for this was that there's been a lot of issues categorizing these other small RNAs based on the regulatory roles they play, the preferred site of action, and their mechanisms. So the study I found tried to do this as succinctly as possible. So one of the regulatory RNAs they found in bacteria, or the two categories they decided to put these in, were the cis-acting RNAs and the trans-acting RNAs. The cis-acting RNAs describe regulatory RNAs that act on the untranslated regions of mRNA, so either the 5' untranslated region or the 3' untranslated region. The trans-acting RNAs, however, are a bit more interesting. They're comprised of cis-encoding antisense RNAs and transencoded RNAs. So these will include like your antitoxin, toxin modules. This will include your tracer RNAs, your guide RNAs, your CRISPR RNAs, and the like. This is a slide just showing a little bit more detail as to the RNA, a little bit more detail about the RNAs that fall into these categories. So this slide right here describing cis-acting small RNAs and their mechanisms of action. For the figure A, right here, the 5' untranslated region mediated transcriptional regulation is the 5' untranslated region mediated transcriptional regulation. On the left, in the absence of ligand, transcription is initiated through a permissive stem loop region. An aptamer region in the 5' untranslated region of an open reading frame. In the presence of ligand, the aptamer region assumes the conformational change and is stabilized by the direction recognition of ligand. The complex leads to the formation of U-rich hairpin that acts as a transcriptional terminator. On the right, in the absence of ligand, transcription is initially attenuated. The recognition of the ligand by the U-rich hairpin leads to the disruption of the stem loop and thus to the transcription, anti-termination, or activation. Below that, for translation, the 5' untranslated region mediated translation regulation. In the absence of ligand, the aptamer region forms an anti-terminator hairpin. The ribosome binding site is available and mRNA translation is initiated. Upon ligand binding, the access to the ribosome binding site is hindered, preventing translation. On the right of that, in the absence of ligand, translation is inhibited due to sequestered ribosome binding sites. The recognition of ligand by the stem loop releases the ribosome binding site, and translation can be initiated. In other cases, the ligand can be replaced by a temperature-dependent mechanism, where the mRNA adopts a secondary structure that hinders the access of the RBS, ribosome binding site. For this mechanism of RNA regulation by antisense RNAs, that's for the second set of RNAs we're talking about. The mechanism of mRNA regulation by antisense RNAs, that is on the upper left. The binding of the AsRNAs to the translation initiation region leads to translation repression. The RNA duplex can be degraded by RNase 3, a double-stranded specific N-ribonuclease conserved in all the three kingdoms, or by RNase E, which belongs to gram-negative bacteria degradosome. On the left lower corner, the AsRNA can bind the 5' end of the target mRNA and cause changes in the target RNA structure, leading to transcription termination. On the right, the complex AsRNA can also stabilize target mRNA, target mRNA for translation activation. And for the last portion, transencoding for transencoded small RNAs. The transencoded small RNAs based regulatory mechanism of microRNA expression. The transencoding small RNAs act through an imperfect base pairing to mRNA targets. The transencoded small RNA mRNA duplex is stabilized by HFQ and can either repress as seen in A and B, activate as seen in C, or stabilize as seen in mRNA translation as seen in D. In E, it's also demonstrated that transencoded small RNAs can bind to proteins to inhibit and/or modify the activity. In F, tracer RNAs are transencoded small RNAs involved in the maturation of CRISPR RNAs through interaction with the complex CRISPR-Cas9 for CRISPR RNAs and RNase 3. The processed CRISPR RNA targets the invading DNA. So what is hidden behind the RNA or transcriptome of S of Streptococcus agalactici as an example? It is typically responsible for neonatal infections like pneumonia and in some cases meningitis. Why this is important is because it lives in extremely acidic environments and possesses a CIRH gene. Several in silico studies have been done on this species that have yielded several other cis-acting RNAs, as well as one that was based on the Rho-independent terminator as a specific signature of the gene loci yielded 197 small RNAs, some of which were found to be activated in response to acid stress. So right here, you can sort of see how important a role this plays in the activation or repression of genes under acid stress, especially for the species being able to survive in highly acidic environments, say like the stomach of an animal, for instance. I remember during the semester we discussed the lacoperon and how drops in the level of glucose, or how glucose acted as a ligand for a transcription factor that activated the gene to start using lactose instead of glucose in this case. Instead of it being a transcription factor, it's an RNA molecule. Here is a module showing how small RNAs also help bacteria. In the event of exposure to a toxin. This figure is particularly useful if one considers how some bacteria are able to sequester a toxin and then synthesize a antitoxin using microRNAs.